This is an audio slideshow about my RET experience with
Sheila Patek, and I'm a professor at UMass Biology Department.
And I'm Janice St. Pierre. I'm a teacher from Bath Haverling High School in New York.
We study mantis shrimp in my lab. It's a picture of one as a mural going up to the lab. And as you can see, Jan St. Pierre's name is on the door sign for our Patek Lab this summer. And we study some pretty interesting creatures called mantis shrimp, or stomatopods.
The really unusual organisms, you can see that they have this appendage that is very armored. It has the ability to break glass in a fraction of a second.
Yeah, they're pretty scary animals, and we have a bunch of them in our lab. And I guess the main structure of our lab is a wet lab, and you can see Jan heading on in the door. And we have a big open space areas for building models and running experiments, and also places for students to work in the lab.
You can see that each day someone has to check in on the animals, and my day was on Thursdays. You go in and check the salinity using a refractometer. You also check to make sure all the animals are alive, and you get to feed them. My favorite one was this one down on the end. She was pretty aggressive, and she's got a really interesting behavior. They always come out to greet you each day when you've got their snail or their mollusk that they're going to eat, and some are a little bit on the shy side.
We also do all kinds of experiments in the lab. We can measure forces that the animals produce, and we also can measure extremely high-speed video to see their movements because they're moving so quickly. So the aquarium room is a major part of our lab where we can actually study the animals.
Here I am working with Mike Rosario. He's a PhD candidate, and he was basically, besides Sheila, one of my major mentors, and we were working on the anatomy of the striking appendage. To start off with, I wanted to know about the anatomy, so we did a dissection, and the whole lab joined in on dissecting the organisms and looking at the structure of the appendages.
It was really fun seeing everybody together on one day. We just had this, we called it Dissection Day. We have a postdoc from Japan, and multiple graduate students and undergraduates in the lab showed up, and we just looked at it and tried to see the morphology in new ways.
There is a very diverse group of people that work in this lab, and it's really interesting to see how the different ideas bounce from a person that had a psychology major now into biomechanics. The integration of all these different fields coming in to solve a problem were very interesting.
It was great to see Jan learning some new techniques on the microscope, and also how to mount an appendage to study the elastic energy storage, and that was her main focus for her research project.
This is the force machine that we looked at, how much energy it took to compress the appendage. The appendage compresses and stores elastic energy, and then it releases it in a strike. Once I did the force machine, I took the results and I had to manipulate the data, and Mike helped trying to analyze the data that I achieved.
Jan came up with some amazingly interesting results and really stimulated us to think about the elastic mechanism in new ways. And because of her time here, we're all thinking about these animals differently, and it's been an amazing month. Thank you very much, Jan.
It has been an amazing experience. Thank you.